Okay, very good. Okay, good morning, good afternoon, good evening, everybody. Thank you very much for joining this webinar today. We are very fortunate to have Dr. Waseem Asghar with us. He's going to talk about a topic which is very interesting for all of us. The topic is, can smartphones be used for disease testing? And he will be telling us how smartphones can be used to test for corona, HIV, and bacteria. Can smartphones be used to get rapid testing at home? Challenges and opportunities in biosensing research. This talk is a result of a collaboration between STEM Matters and Young Professionals Society. STEM Matters is an organization here in Dallas, Texas. Our vision is to enable students to innovate, invent, and deliver quality solutions to contemporary challenges. And our mission is to inspire students to excel in the areas of science, technology, engineering, and environment matters by nurturing their critical and analytical thinking skills. We have objectives that we want to accomplish as part of this organization. We want to nurture highly functioning teams to work together and create significantly sound, innovative solutions to the world's complex problems, create a community of young innovators trained in problem-solving skills, develop high-level scientific programs to provide an international forum for scientific contact, to learn, collaborate, and compete for meritorious outcomes. We empower students with the skill sets, for example, research, programming, leadership, and others, to make them competitive in the higher education and marketplace. We involve underprivileged youth in science, technology, engineering, and environment matters. Our sister organization is Young Professional Society from UK. It is set up to help support and change the lives of youth by providing personal and professional development opportunities. It aims to bring to the forefront a more progressive, accomplished, and enlightened younger generation of people who will be the representatives and leaders of their communities and who will contribute positively towards the wider society. So we are very happy to have Dr. Asghar with us. He earned his PhD from the University of Arlington, Texas, and currently he's an associate professor at Florida Atlantic University. He's a biomedical scientist and a nanotechnologist, winner of the NSF Career Award, Humanity in Science Award, FAU's Researcher of the Year Award, ASEE New Faculty Research Award, and FAU Mentoring Award. So we have a very accomplished person with us. We greatly thank Dr. Asghar for being with us today. Dr. Asghar, over to you.
Sure, thank you. It's my pleasure actually to talk with you guys. I presented, I think, a couple of months before also to the STEM Matters. I think it's a great initiative, and I always love to talk to the people. So somehow I think today we don't have so many people, but last I remember there were like 20, 30 people online. So I think it's maybe a Sunday evening, so that's everyone is busy with their stuff. Or maybe in the United States, we are going on a Halloween, so people may be busy with that. So, but anyways, thank you for a very nice introduction. My background is not that great, you know, so I'm sitting in a room where there also sunshine comes in. So sometimes you see there is a shiny thing on my face, but I cannot really get rid of that. I hope it's fine. Okay, so now we'll continue. Just go ahead with the presentation, and then we'll talk more about different projects which I am working on, more specifically related with the use of a smartphone for disease testing. Okay. As Mr. Shakil mentioned, that I'm working as an associate professor in the Department of Computer and Electrical Engineering and Computer Science at Florida Atlantic University. And I'm also a director of a research lab. It's called the Asghar Lab, Micro and Nanotechnology in Medicine. So today's topic will be whether we can use smartphone for disease testing. So a little bit more about my background. I did my undergrad from Pakistan. Then I did my PhD from University of Texas, Arlington, in 2012. And then I moved to Harvard, MIT Health Science and Technology Division. I did my postdoc there for 2 years approximately. And then I spent then I moved to West Coast. I spent about 6 months there at the Stanford School of Medicine to work on different areas of biomedical engineering, more specifically disease diagnostics. And from there, from 2014, I moved to Florida, Florida Atlantic University. That's the building where I'm right now located. So our department is housed in this building. It's pretty nice and new building. It's called the Green Building, means we can make this building bright with the less energy. So that is designed, and that's how it's designed. So it's very nice building, which was not built very long time ago. So that's where I am, and I've been working now at Florida Atlantic University from last 6 years. Okay, now we'll talk a little bit about the background. So what we will be looking for. So basically, if we look at just the scale factor, where the things are. So I know there are some high school students who will be also watching this lecture. So sometimes I will try to make it things a little bit simpler so that people can follow. And sometimes things are a little bit abstract for maybe high school students, but we'll see how it goes. Just in case if you have any questions, I think you can just turn on your mic and ask the question right away. And we will also have a question answer session at the end. That is also fine, but you can stop anytime if there's any question. Okay, so if we look at the retinal implants, so they are in a micrometer scale, means they are very, very small. I think most of you have an idea what millimeter is. Then we have some devices which are even smaller, on smaller scale, like a micrometer scale, which is 10 raised to power minus 6. So if you look at our human hair, and the thickness of a human hair is about 40 to 50 micron of one hair. So from there you can get an idea what scale factor we are talking about here. So one of the project today, which I'll be discussing, I will be talking about a bacteria detection. And bacteria, their diameters are also in the range of 1 to 5 micrometer. So they are very, very small. So in terms of thickness, they are about 10 times thinner, or their size factor is 10 times smaller than the thickness of a human hair. So now you can think about, if you really want to see bacteria, you need some kind of a microscope or some other biosensor, so because you cannot really see those bacteria with the naked eye. And then we even have some materials which are synthetic, and also some natural materials which are on a nanometer scale. A nanometer is actually 1,000 times smaller than the micrometer. So when we talk about nanometer, so this is a buckyball, and these are the carbon nanotubes. So their diameters are in few nanometers. But if you look at natural materials, we have human DNA or RNA, which is, if you look at the thickness, it's about 1 to 2 nanometer thick. So if from left to right, if you look at this ladder, so it's about 1 to 2 nanometer thick. So it's very, very, very actually small structures or materials which we like to focus on for biosensing purpose. In today's talk, we'll also talk about the viruses, and so the virus size is around 3 to 50 nanometer. So this is not actually correct. Some viruses are larger than 50 nanometer. For example, HIV is a 110 nanometer. Zika or dengue is about 50. So yeah, their range is in nanometers. Basically, it can be either 100. So I think most of the viruses fall into less than 150 nanometer. But they are still in nanometer range. They are very, very small. We cannot see them. So detecting them becomes very, very difficult. Nowadays, everyone knows about the coronavirus. It's a pandemic is still going on, and the virus sizes are also less than 100 nanometers. So they are very, very small, and detection is very difficult. We cannot really see these viruses under the microscope very easily. So I remember in the United States when this all started, we have a big backlog about the availability of the tests. So whenever even the hospitals, they were doing the testing, so but people have to wait more than 10 days to get the results. By that time, things are not the same when they had the infection. But now things have significantly improved in diagnostics as far as the coronavirus is concerned. But still, it takes a couple of days basically to get your results back, and it's not that handy. So you have to really go and set up an appointment, go to the diagnostic facility and get your nasal swab done, and then after a couple of days you'll be getting your results. So that's where we are. I'm talking about the United States, and we are in Florida, where we have all the facilities, but we have many different places around the world, even in the United States, some suburbs where these facilities are not easily available. So then it takes much longer in those places. And in developing world, in other parts of the world, these things are even scarce. And so getting the detection is a first step, actually, if we really want to manage this disease better, because once we know that someone is infected, then we can do the next steps. Like we can ask that person, we can isolate that person, or we can actually start doing some treatment. So the first thing is detection. So that's very, very important. Okay. So you might have seen that there are some point of care biosensors already available in the market. For example, you might have heard about pregnancy tests, which are lateral flow assay strips. Like they are just paper strips where if someone urinate on top of it, and if the person is pregnant, and their HCG hormone level is much higher, and then you will see actually a one line made up of actually gold nanoparticles. You don't see those nanoparticles, but there are gold nanoparticles there which aggregate and make up a line. So that's how you see that if someone is pregnant or not. We also have other devices available, and other very commonly used devices, glucose meter, the one on the top left. So that's also very handy. Previously, I remember when people want to get their glucose level checked, they really have to go to blood work. They have to do the blood works, and it takes a couple of days, and you have to set up a time. And by the time you get your results back after a couple of days, your glucose level is not the same. So you cannot really do much at that time, because it keep on changing. So for management of a disease, like because some people are also taking insulin, which is used in therapeutics, and you need to know how much is your glucose level right away before injecting insulin. So with the invention of this device, these things has much more improved the healthcare, specifically in the diabetes. So that's very, very handy. So, but we don't see these devices available for many other diseases. There are tons of other diseases, like viral diseases or bacterial diseases, where we don't see those devices. They are not available. So the part of the reason is that the sensing mechanisms are complex. When we work with the blood sample, they are complex. Sometimes those viruses or bacteria which we want to detect, they are present in such a small quantity that we will not be able to actually see them or easily detect them by the use of these devices. And that's why you don't see these things of like point of care diagnostics for many other infections or diseases. So now in today's talk, we'll try to explore if we can use smartphone basically to detect some of these diseases. Why smartphone? Because smartphone is kind of a computer. So it has a microprocessor, it has a very nice camera, which can be developed into some kind of a microscope. And you can also develop some algorithms on the smartphone, and it can be used for many different applications. It can be used for contact tracing, means that's for coronavirus, for example. If someone is infected, so we can really hypothetically track that person, where he went, and whether he has made a contact with any other person. So there are a lot of groups who are working on those areas. So my thought was, okay, why we can't build some devices around a smartphone? Because a lot of people nowadays have a smartphone, many of them. So if we make something on a smartphone, so that can be ideally be made as a point of care device, or you can actually think about having a home-based diagnostic devices which you can use a smartphone. You can attach the device with your smartphone, and you can test different diseases. So let's go through some of the projects which I have been recently involved. So the first project I'll be talking about will be about sickle cell disease. So sickle cell disease is a disease of a human red blood cells, and it's because of some variations, mutations in the autosomal recessive inherited disease. So it's some of the variations, one of the gene of a human body, and then it's recognized by WHO as a public health concern. In Africa, about 25% of the people have the gene for sickle cell disease. And in United States also, there are tons of people who have this disease. So, but the situation in Africa is very, very severe. And so what happens if someone has a sickle cell disease? So in a sickle cell disease, your red blood cells actually doesn't retain the same shape how they look like normally. So normally they have a donut shape. It's about 6 micrometer in diameter on one side and about 2 micrometer in terms of thickness. So it looks like a donut if you see it under the microscope. But if someone has a sickle cell disease, their shape, there are some polymerization which goes on with the inside the red blood cells, and then the cells actually have a weird shapes, and also they lose their flexibility. They become very rigid. So those cells actually also die quickly, and they cannot efficiently transfer oxygen to different parts of the body. So that's a problem. And as you can see from these numbers that the situation is really bad in Africa. On the other hand, now we don't have any high-tech laboratories available in Africa. So the diagnostics is not that very straightforward. And also some people take drugs to manage the disease. In that case, we also have to look at what is their what is the quantification of red blood cells, like how many red blood cells are sickled at this moment, whether these people should take this drug or not. So some test which can be performed at home at a point of care, where people can see their sickle cell disease status before taking therapeutics over a period of time, so that will be very, very helpful. And we don't have anything right now in the market. So this is another graph which tells you how severe this disease is. This data shows from 1910 to about 2000, and you can see the life expectancy of all Americans, which is about a little bit less than 80 years. But now people who have a sickle cell disease, their life expectancy is still less than 50 in 2000. So right now it's also around 50, but you can see that with some interventions, with the focus of research teams, there are some new therapeutics, so that people were able to actually live longer as compared to how, like in 60s, mostly people were dying in less than 10 years of age. So things are getting better, but we really want to make sure that it goes further higher so that people can live longer with this disease. So when we look at red blood cells, so that's what I was mentioning. So there are cells which are donut shaped. These are the healthy cells, but there are some cells which doesn't retain those shapes, so they become elongated and they become also rigid. So those are called the sickle cells. Their surface area becomes smaller. They cannot absorb enough oxygen to transfer. So we have also done some experiment in our lab where we actually look at how sickle cells behave when we expose them to an environment where there is less oxygen. So in that experiment, if you see the small video, so these cells were actually fine before, and then we removed oxygen from the environment, and this is a microscopic image. You can see cells start shrinking right away. So in a few seconds, they start shrinking, and that's what happens when cells are sickled. In a normal cell, if the red blood cell is normal, it actually retains its shape and it can transfer the oxygen. Okay, so basically there is a need to develop a portable or some cost-effective, user-friendly device which can be used to look at these cells and see if the cells are sickled, or if the cells are sickled, then how many cells are sickled out of, what's the percentage of cell which is sickled. So this type of a device will be very, very helpful to manage the disease over a period of time. So what we thought, okay, we have a smartphone. Almost everyone has a smartphone. So can we think about using a smartphone and convert it into some type of microscope, and then integrate it with a device which can have a small drop of blood, and then we can look at those cells and see if the cells are sickled or not. But in this whole setup, we also have to create an environment which doesn't have a high concentration of oxygen, so that we can see cell sickling very rapidly. So to do that, what we have done is we took a smartphone. We actually attach some external lens with it, with the camera of a smartphone, and then we also built a small black box which can hold one slide, and in this slide we have microfluidic channel. Means there is a small channel where you can put just one drop of blood, and it spreads very evenly on the surface of the channel, and then you can see if everything is designed properly. So we also have light source and other things are also there. I will not go into those details, but you should be able to see those cells. So this image actually shows you how the cells looks like, red blood cells looks like, with the cell phone, and this is not possible if you don't have the lens attached with the camera of the cell phone. And then you also have to maintain specific distance, how much far it should be, because if it's not at the focal length, then you don't really see those cells very clearly. So once we build that, cell phone setup, and then you have an app where you can see, okay, how many cells are sickled or not. So we have to develop some algorithms so that we can quantify the cell shapes. Then now to look at the blood, we cannot look at the whole blood itself. We have to spread the blood very evenly on the surface. For that, we have made a very low cost, very low cost device. Means it's all built up with the help of just plastic sheets and adhesive, which is a double-sided adhesive. So you connect by using laser cutter, you cut them into channels. So you make this type of a device, and this is a channel basically where you will be putting a drop of blood. Even less than a drop of blood can be put in there, and it will fill the whole channel very easily. So once the blood is there in the channel, and the height of the channel is 50 micrometer, it means 25 to 50 micrometer. So it means you have hardly one layer of the cells which will be in the channel, because if there are multiple layers of the cells, you will not be able to see single cell. Okay, then what we did is we just take the image with the help of a smartphone and application, and then we did quite a few steps to clean up the image. What from cleanup means, basically you take the image, convert it into a grayscale, also remove the noise if there, because in cell phone the problem is that one time you take one image, you get a very nice image. Next time you may not be getting that type of an image if your background is not consistent. So that is one of the major challenge when you use the cell phone. And so for that reason, we actually built that box, so which makes sure that your background is almost consistent all the time. And then we have to remove the background. We have to do some water shedding and segmentation. At the end, we should be able to actually quantify how many cells are round, or we would define which cells are round or which cells are not, and we should be able to see the non-round cells. This is a little bit of a magnified image of how the cells looks like. So this is the image when the cells, this is how the red blood cells looks like. And with the image processing, this is after the image processing, you see the black and white. So white are the cells. And then we quantified. So in this sample, so all these cells were considered normal, and these cells are not normal. So they are non-round or sickle cell, most probably. Then we have another image where we have done some treatment by a salt, which can consume all the oxygen from the environment. So that makes the blood sickled, basically. if the blood is healthy, then the cells will not be sickled. They will retain their shape. But if the blood is from sickle patient, then you will see that the cells will start having weird shapes. And at the end, we were able to see actually a lot more cells than the normal case. That many cells were actually have very different shapes, and this all can be done in less than 10 minutes with the help of this device. So then we do some patient testing. So if you look at on the top of this, it's a big table, but I will just point you to certain areas. So this is the normal blood. First image is when we use our smartphone based setup, and the second image is when we use a microscope, like lab-based microscope. So in the case of normal blood, we were able to see about 0.86% of the cells which were sickled, which is normal. At least few cells may be dying, or they may have a little bit different shape. So this is normal. And in the microscope, we were seeing 0.2% of the cells. So this is within a close range. But so it means our setup and the lab-based microscope setup, they were reporting very similar results. Then we also did some treatment, and also, on the top we have another sample from another person, which was normal, and then we have 2 patient samples, which it means they have confirmed sickle cell disease. So if you look at those patients, so in our setup we were reporting 6.3% as compared to 6.14% when we use a standard method. So it was very close and very correlated results. And for second patient, we were getting 3.7% and 3.4% by using both different methods. And then we did some treatment. Treatment means there is one channel which has a hypoxic environment, means it has a very, very less oxygen. So if you look at those images, with our setup, we were getting about 43% of the cells become sickle in the case of a patient, and only 40%, 40.5% of, and also the 40.5% cells were seen sickled when you use a microscope. And same is the case when you use the other patient sample, as compared to the normal sample, where you get actually 2.9 or 0.45% cells. So basically, this is significantly more cells sickled, and the numbers which we were getting were very consistent as compared when we compared with the standard method. So basically, by using this cell phone based setup, you can do all this quantification of the cells, and you can create an environment in a microfluidics which will be hypoxic, and you should be able to see actually cells with this setup. So this was very interesting work, and we got a lot of appreciation from people around that we can convert this smartphone into some kind of a microscope, and then you can integrate that microscope with the microfluidics, and also you can create some hypoxic environment. So far, we have done the validation with 2 normal samples and 2 patient samples, and results were very consistent. So we are still working on this project, and we are now in the process of actually validating this device with a lot more patient samples and healthy samples. So the second project I want to talk about, so that was all about sickle cell. So the second project I want to talk about is detection of bacteria. So bacteria is also, some of the bacteria are harmful. For example, some strains of the E. coli is one type of bacteria. They're harmful, and several of us, at least once in a year, we have some bacterial infection where people take antibiotics and all that. So bacteria, some strains of bacteria definitely cause a different type of diseases, like people have a diarrhea. There's also chances of sepsis, means when the bacteria infection goes out of control, then it creates sepsis, which has a very high mortality rate. So if someone has a sepsis, very difficult to then control the disease at that time. And also, if the food is not in good quality, food can be also infected with the bacteria or fungus. So that can also create a lot of disease in the US and all around the world. The problem is that why we don't test the bacteria very often. And the reason is the current testing methods take about a week, or at least few days to get tested. So what happens if someone is infected? normally in many parts of the world, people just prescribe antibiotics. In developed world, uh, the doctor suggests, okay, you have to have the bacteria testing or or fungus testing, and normally that takes about a week. By the time you have the results back, if the symptoms are not recovering, they also prescribe you antibiotics. So by the time the results are back, you already have uh, you already have cured, and then no one actually worries about if it was bacterial or whether it was a viral infection. So the reason is that the testing protocol is so long; it takes forever actually to test if someone has bacteria or not. Uh, then so the tests doesn't have much value, except in certain cases where the infection is not going away. Then we want to make sure that we test that person for bacteria, and we have to prescribe uh, relevant antibiotics and the dosage of antibiotics. But because there is no rapid testing available, and because of that, uh, people normally take antibiotics uh, about at least 50% times when they don't need it, and that creates some resistant strains of bacteria, which is another problem. So, but without going into those details, I just want to highlight that the current methods of bacteria detection, it can be either food-based bacteria infection, or it can be, some people who have a kidney diseases, they go through some process called the peritoneal dialysis. Basically, they do dialysis, they have one bag always hanging around with their belly, which has a waste fluid. So sometimes those people also have infections, but there is no such way that you can identify the infection quickly, because the current testing methods take forever, and normally it's not easy to detect bacteria by using the standard methods. So then what we thought about in this work, that we thought about, okay, let's think about gold nanoparticles, for example. So we have noticed that the gold nanoparticles, when they are present on different sizes, they actually produce a very different color. For example, if you look at this top image, all of them are the suspensions of the gold nanoparticles. So these are the smaller ones, I believe 5 nanometer, and this is 50 nanometer. So as the gold nanoparticle size increases, the color which you see, so basically their absorbance changes. All this is the same material, but changing on the color, how it interacts with the light, a different size scale, you see a different color of those suspensions. Okay, so we thought, okay, maybe we can have some gold nanoparticles. We can functionalize those gold nanoparticles with some antibodies or proteins which can bind very specifically with the bacteria. So let's say if the gold nanoparticles are functionalized with some proteins which can bind with the bacteria surface, so then we can think about if we now mix these nanoparticles with this in a solution which has a bacteria. So these particles go there and bind on the surface of bacteria, if there is a bacteria. So the bacteria will aggregate. Basically, you will see a lot of particles there sitting on the surface of the bacteria, and then they also aggregate, because these particles can bind with the other bacteria also. So, and then when these particles aggregate, their effective size, although they were 15 nanometer, for example, but once they aggregate, their effective size or how they interact with the light uh, would be larger, and then you would be able to actually see a different color, because this is the property of the particles. So these are some scanning electron microscope images, shows you the real images of the bacteria, once how they look like when they are aggregated, and you can see it here very clearly. And this is the image where you see that in a control where we don't have a bacteria, you see a pinkish color of the gold nanoparticles. But when there is a bacteria, you see some aggregation there, even with the naked eye, and also see the color is a little bit bluish, and that's what was expected. So then what we did is we designed a smartphone-based app where you can take an image of this test can be done actually on a paper. You can put a just drop of the mixture of the bacteria and the particles, and you put it on the paper, white with a white background, and you can just take the one image. And with the smartphone, you can quantify the red, green, blue value, or 1 minus the red value. It depends on what parameter you want to look at. But basically, you can quantify the intensity or the color of those solutions. And so what we did is we spiked bacteria, means we mix bacteria in different type of solutions, including PBS, which is just a regular buffer, which is like a salt mixed with the water. And then also we mix it in the serum, and also we mix it with the peritoneal dialysis fluid, the fluid which I was mentioning when people have some peritoneal disease. So, so in those cases, we noticed that whenever there is a bacteria, we were able to see actually aggregation, and that is quantified. For example, in here, when we have 10 to the power 6 bacteria, the value which we are getting from our algorithm was 0.6. And when we have a less number of bacteria present in a solution, you get a much smaller value. So basically, you kind of get a linear response. In this case, we're 0.76, which is not that great, but at least you can detect if the bacteria is there or not. And you can, to some extent, you can also quantify how much bacteria was there. So that was a very straightforward solution of doing the bacterial detection, and we have reported these results previously. So, but in general, this method works pretty well when you have a bacteria which are isolated from the raw sample. So raw sample means it can be either food sample or it can be a blood sample. But this method cannot be really applied in the field right away. The reason is that when we look at the sample, it's not an isolated bacteria. It's either in the blood or it can be in the food or in any other solution. So that creates the challenges, and also the opportunities. Means somehow we have to develop methods where we can quickly isolate those bacteria by using some sample prep, and we can remove the background. Means we have to isolate those bacteria very rapidly, and once it's isolated, then this type of sensor can make sense, and it can detect the bacteria from a cleaner solution very rapidly. But at least that's the first step towards developing some point of care sensor which can be used for bacterial detection. Okay, now we'll move on our third project, which will be a last one in today's presentation, and then we'll also talk about the coronavirus detection, whether that is possible or not. So we have been working on Zika virus, which is a flavivirus. Basically, it is transmitted through mosquitoes, some different type of mosquitoes, and you might have heard about also dengue fever. So the dengue fever is also happens because of same mosquito, and dengue virus is kind of a brother of Zika virus because they belong to the same family. They both are flaviviruses. So in Zika in 2016, the world has seen a large number of people who are like new babies which were born. They have a smaller heads, and that disease is called the microcephaly, as shown here. So their head size becomes smaller. So it's a congenital disease, means their brain doesn't grow larger or like normally. So and they are delayed kids. So there were 5,000 such cases were reported in Brazil in 2016, and that was scary. And then they figured out, okay, that happened because there was a Zika infection. Previously, people didn't know about it. So, and then all of a sudden, then we start testing Zika at different places, and we also start looking at the spread of the Zika. And at that time, I remember in Florida also, there were a high number of cases with the Zika related, but luckily that disease actually kind of the spread kind of reduced now, but it can come back anytime. You never know. Based on these diseases come back, they come on and go away after a few years. Like in dengue, normally it comes to South Asia normally in a winter season, and then goes away normally in a hot summer. So but that's how that virus behaves. But I don't think the coronavirus is behaving a bit differently. But anyways, so with the Zika, the problem was that this was a big issue because you can have a generation where many people may have a small head. So that's what we don't want. And this virus have flu-like symptoms, like people can have a headache or rash or fever like you have with the other viruses. So it's very difficult to actually identify whether it's because of a Zika or maybe flu or some other virus. So and there are also dengue and chikungunya virus, which are also flaviviruses, and then it's very difficult to distinguish whether this infection is from Zika virus or not. But the problem is that normally, if you're a male or in general a healthy person, if they have infection, it goes away after a week or 2 weeks, like a flu. But if someone is pregnant and if the lady catch up this virus, so what happens? Then if there's a newborn, the newborn will be severely affected because then virus doesn't go away. It actually infects the fetus. It infects the newborn baby in the belly, and once the baby is born, and that baby will have the microcephaly type of diseases, and that's not very good. So that's why it's very important to identify if someone has a uh, you know uh, the Zika infection, and also then you can guide those people that okay, stay isolated, or if someone is planning to have a baby, so maybe they should wait for the infection to go away. So those type of things can be done, but detection and looking at the spread of this disease is very, very important. uh, so for detection, again, for virus detection, normally the standard methods are lab-based. They're expensive. They're not easily available. You cannot detect those viruses at doctor's office or at home. So that was an issue. So then we thought about actually designing some type of a platform which can be rapid, which can be actually used at a point of care. And all of these things, which I'll be discussing from now onwards, they can be equally applicable to the coronavirus detection. So the standard method of detecting the uh, most of the viral diseases is based on the process is called the PCR, polymerase chain reaction, basically. So you first, let's say you take a blood, you isolate the viruses with the process, and then you lyse those viruses. It means you rupture those viruses, you remove the RNA or DNA from the virus, you again isolate it, wash it, and then you perform the PCR process. So PCR requires a thermocycler. So it's a big equipment. It's very expensive. Tens of thousands of dollars is a cost. So it's not very easily available in the developing world. And the tests are also costly. So one test will cost at least $50 to $100. So it's not very useful for the developing world. So we are definitely looking for ways how we can identify these viruses much rapidly, ideally at the point of care, and also the test has to be low cost. So right now, if you look at the process, it takes much longer also, at least 4 or 5 hours. And you need a trained technician, you need a fancy lab to run these assays, and the chances of contaminations are high. So yeah, definitely there's a large room of improvements which are needed. So what we thought about, okay, whether we can do all this process on a one device which we are making, whether we can do all this processing on a single device, whether that's possible or not. So, and then somehow we want to get rid of this thermocycler, which is a type of equipment which keep on changing temperature, because that's the requirement of a PCR. There are alternative methods. So what PCR does, actually it amplifies the RNA or DNA of the virus. So if there's a virus, there are 10 copies, so it amplifies it to maybe 100 copies, 1,000 copies, 10,000 copies. So it make it a large amount, a quantity of those copies of the RNA or DNA of the virus. So then when you have a higher number of copies, then you can see those viruses very easily with the help of some dye, or with the help of some nanoparticles. So there are many different sensing mechanisms which you can use once the viruses are present at a very high copy number. But in a raw sample, they are not present at a very high copy number in many times. So that's why we always have to use some type of a method to amplify the viruses, because seeing viruses is not very easy. So you can amplify their RNA and DNA. So, and then once they're present in large quantity, then you can see them easily. So what we thought about is designing a device where it will have different chambers, and it has to be also low cost. So each chamber is actually isolated. So there are some chambers which are which are aqueous, and there are some chambers which have oil in there. So the elliptical chambers here shown as valving chambers, it has a mineral oil. This also has a mineral oil, and the chambers shown as a bluish or the red or green or the chamber one. So these are the chambers which are aqueous. Basically, you can put in a blood in chamber one, and then we put in some magnetic particles, nanoparticles there, which can move, which can bind with the RNA once the virus is lysed, and then it moves it to the next chamber through this oil, and then in next chamber we do some cleanup, and then and move to a third chamber, second round of washing to do the cleanup, and the last chamber we do the amplification. For amplification, we use another process called the loop-mediated isothermal amplification. Basically, in simple words, this amplification doesn't require thermocycler. It just requires one temperature. For example, you have to maintain certain temperature. In this case, it was about 70 degrees centigrade. So if you maintain that temperature on the chip, you should be able to actually do the amplification. And so in order to achieve all this, we build one setup which is not very big. And you can, this is kind of a prototype of the platform which we have developed. And once you really want to sell it or you commercialize it, you really have to make it a little bit more nicer in terms of cosmetics. But basically, in general, if you look at the platform, it has one motor. We have magnets under the chip. You can put the chip on the magnets, and the magnets actually move those nanoparticles from one chamber to the other. And all the processing can be done in 9 minutes, basically. And then for amplification, it's another 20 minutes. So basically, in around 30 minutes, you should be able to see if there is any virus in the sample or not. So the good thing about this assay is that you don't really have to clean up the sample before. So this process itself will clean up the sample. Basically, it can isolate the viruses from the blood sample or plasma sample, and then it can do the other processing there. We also have some electronic components there to control the movement of those magnets. We use a motor, and also we have a heater which is attached on the surface, so to maintain certain temperature. And the heater is also controlled with the help of microcontroller, so you can maintain certain temperature on the chamber of the device. So I'll show you a small, yes?
We have a bunch of questions. Do you think after this short video, we can get into the questions?
Yeah, that should be fine, I think. Yeah, I think I'll be wrapping up now, I think in the next 5 to 10 minutes. And then we can have a question and answer session. How does that work? Is it fine?
Yeah, from a timing perspective, I think we're 10 minutes left. So I was wondering if we can get into the question and answers after this short video that you are about to play.
Okay, so just give me, I think, just a couple of minutes. I think at least 3 minutes, and I'll try to wrap it up. I think we are very close to finish.
Very good.
Okay, so now let's look at this video. So in this video, this is a before image, how the sample look like. And you see, this shows you when we have the platform in operation, it moves those particles, and it actually shows you those magnets moving those particles from one chamber to other automatically. So this whole thing can be done automatically. And at the end, you have some dye which will generate a different color. So in this case, it was bluish. So when you have a Zika virus, you are seeing actually blue color. Next, we look at another image where we looked at the sensitivity, like how much small in number we can detect. So we were able to actually see 10 power 2. It means if there are 100 copies of Zika in 1 ml of blood, we were able to actually see it, and that's why you see the different color. Others where we don't see a color, either it's a HIV or it's a control where you don't see a color. Then we tested this whole platform with some clinical samples, and we were able to see in the clinical samples that all the samples which have Zika here, they were showing the amplification, and also it can be seen in this case of a gel. Like only Zika shows amplification. The other controls which doesn't have Zika virus, or dengue was used as a another virus, it doesn't show anything. Basically, this test was very specific, and we also performed this test on a platform, and it shows you B, C, D, and E shows that there was a virus there. and all other A, F, G, and H samples doesn't have a virus. But if you can see from here that it's not very clear that if the color was not very uniform. So sometimes you can do a mistake while identifying whether it's a virus or not. So for that purpose, we thought about using a smartphone. So then we develop another app where we can actually image the whole device with the help of a smartphone. And we noticed that instead of taking one image, you can actually record a video of your platform. And when recording a video, we noticed that there are certain frames where you see a very high actually correlation. Means if you look at the standard error, so the standard error keep on changing based on from how you take the image. So we thought about designing a very novel method of, instead of taking an image, we recorded a small video of 20 seconds, and then the algorithm automatically identifies the frames which are more relevant, and the frames which are noisy, they are removed from the video. So in that way, you can actually build an algorithm which can identify if the color which you are seeing is really showing a positive detection or not. And so then we defined some threshold, and all those 4 samples which are shown on the left side, so they were not, doesn't have the virus. The one which are shown in a circle, they had the virus. Okay, so basically this device we are still working on can be used for coronavirus detection. And so some background about the coronavirus, I will not go into detail because of a time constraint. But basically, coronavirus is also an RNA-based virus. The same method which we developed for Zika can be used for coronavirus. The current testing methods takes much longer time, and we can develop something like we have here, where the smartphone can be used for imaging and analysis, and this type of a platform with the magnetic actuation. And you can have a cell phone to take the image of the device, and you can quantify if someone has the coronavirus or not. And this can be done at a point of care or at doctor's office. So that's what we are doing nowadays. We are working on a coronavirus and developing this test into a new coronavirus rapid testing method, and that's an ongoing project in our lab. Okay, so briefly about my lab. So these are some people who have contributed to some of these projects. So Mazhar, he is my PhD student. He graduated. Actually, he's also listening to today's presentation. So he now is working on a postdoc at the University of Pennsylvania. But other people, they are still working. Some of them are close to graduation. So these are some of the collaborators and some of the funding sources, more specifically NSF and National Institute of Health, Florida Department of Health, and also Fertility and Genetics and the Humanity and Science Award people. So these are some of the organizations which have supported our work. Okay, so now we are good. So we are done, and thank you everyone. And if you have any questions, I'll be more than happy to answer.
Great, very good. Thank you so much, Dr. Sir. That was very insightful. Greatly appreciate your details and very, very interesting projects that you shared with us today. A few questions in the chat box. The first question was, do the negative effects of mobile phones outweigh the benefits? I think the question is coming from use of or sometimes abuse of the cell phone. So how would you respond to this question?
So if I understand this question correctly, it means, are there any limitations of using a cell phone? That's what you're saying?
Yeah, I mean, you mentioned actually the use of the good uses of the mobile phones. And I think the question was, well, these are great uses, but there are a lot of challenges with the cell phones. So any thought on that?
So there are a lot of challenges with the cell phone. That's correct. Basically, when you use these cell phones for diagnostics, we want to make sure that the tests are very repeatable. So what happens when you take an image of a certain assay, and you want to quantify whether this shows infection or not, but the cell phone image can have a very variable background noise, and it's clear, when you take your photo outside under the sun or inside the room or under the light, you always have a very different type of exposure. So some photos are very nice, some are not, and sometimes you get a blurry photos, right? So that all is possible when you use a cell phone, and that limits their use when you really want to develop something with the help of cell phone. But yeah, that also brings a lot of opportunities for the young and old researchers basically to solve those issues. So one effort which we did was to actually recording a video of from different angles, and then select some frames which are actually focused, and so it can remove some of the issues. Did I answer your question or not yet?
No, that's great. If you are okay, I can move on to a couple of other questions. We have a lot of interesting questions for you today.
Okay, please go ahead.
Okay, so what has been the biggest achievement in the field that you're in, that you, for layman people to understand the things that are happening in your field?
Oh, so far.
Yeah. I think before Dr. Asghar goes on to answer this question, just like to inform him that we have some youngsters here who are joining us as well, like from the UK. So some of the questions might be coming from youngsters, in high school or sixth form and that.
I understand. Thank you. So it's very common when we develop these technologies, we also file a patent, means because they have a commercial value. So most of the work which I have shown is now under patent, and one of our previous patents have been licensed by a company, and they are using it in a clinical setting. And so these technologies which I have reported, basically many of them can be developed into the products. So one of our previous device from our lab is now FDA approved. So that's one of the biggest achievement, I would say, in a sense that FDA approved means you can use it in a clinic, and you see, very much satisfied when you see, if you do some work and people are using it. So it's actually for the betterment of the humanity. So all these things which we have discussed, if you really make something interesting for the people and it's useful, so you're also serving the humanity. So I look at it from that angle. So there is also a commercial aspect, and also you want to make sure that you use your capabilities and do research for the betterment of the everyone, basically. So think about it. If you can develop something like a rapid corona test, for example, and if there's some test available, people can buy from the pharmacy and they can use it at home. So that type of availability of that type of a test will drastically improve how we manage the disease. So all those things are definitely encouraging, and I would strongly suggest the young researchers to look into those aspects.
So, Dr. Asghar, what would be your advice for young folks who want to get into nanotechnology or biomedical engineering? What kind of path they should take to get into this field?
So I think when you're in a high school, or if you're not in a college yet, so it depends on, again, I think the first starting step would be to start working, if the possibilities or openings are available, to start working with some research group. I have been mentoring some undergrad or high school students over the past few years. So, and normally if you, for example, if some of you are interested in nanotechnology, you should look at, okay, what are the local good universities where you are living, and then look for, people who are working in these areas, like nanotechnology or bioengineering, if it is of your interest. And by looking at their profile, then contact them and see, okay, if you can volunteer in their lab. So that would be a first step. And when you spend, 6 months in their lab, you will be exposed to a lot of different techniques and a lot of different projects they are working on. And after 6 months, you will be in a position to decide whether you want to really move on with this lab or this type of research, or it may be something which you may not like, basically. There's also a possibility. So that you cannot do that unless you start working in a lab. And if you do some volunteer in 1 or 2 labs, actually that improves your resume also a lot. Doesn't matter where you go. Later on, you may end up going into a medical school. You may end up going into a traditional, engineering degrees. That's also possible. But I would strongly suggest the students who are interested to explore the opportunities with their local universities and try to spend some time in research labs.
Great. We have a couple of questions on Facebook as well. One question, I think, about your first project was, what oxygen scavenging buffer do you use, and microscope images were taken in what mode?
So microscopic images were taken in what mode? I didn't get that. So it was just like a regular mode which we use with the cell phone. Like you take the smartphone, you just take the image of like you take your photo. So that mode we used. I don't know whether that's what is asked. And about the chemical is the long name. So I can actually share with you maybe in give me 1 second. So I can share with you a paper where we have, the chemical which we use for creating that hypoxic environment.
And while Dr. Asghar is pulling it up, my request to the audience is please share your feedback. I have posted the URL in the chat box. Your feedback will greatly help us to figure out what kind of topics you're interested in, and we will be inviting those renowned experts in the respective fields to give talks on the topics that you will suggest. So your input will be greatly appreciated and will greatly help us to come up with the right kind of talks and webinars.
I shared the PDF of the paper, which has more details about the chemicals used and also how we make this device and platform in the chat section. So you can just click there and later on go through it. So it has all those details and also the chemicals.
Okay, I think it was probably mood means TIRF or epifluorescence. I think that was probably the question. I misread it.
Sorry, can you please say it again?
The person who posted the question, he explained mood means TIRF or epifluorescence.
Oh, it was bright field images, not epi, not total internal refraction. It was a bright field image.
Okay. And the other question is, what is the detection limit of the device made to detect the Zika virus?
It was 100 viruses.
Okay. Another question is, how long will it take you to develop a smartphone-based diagnostic test for COVID-19, and how accurate can or will it be? What would be the cost for such a COVID test?
So one by one. So the cost for such a test would be about $5 roughly. How long it takes to develop such a test? Okay, that's an interesting question. The problem is that the reagents which are used to develop such a test are not easily available nowadays for researchers. Basically, for my lab, if I order something related with the coronavirus, it takes about a month to get that thing to the lab. That slows down the things because a lot of commercial entities have been involved in developing some assays, and the companies have a privilege actually to get those research material. So in an academic environment, things are a little bit slow as when we talk about the coronavirus related reagents. So if we have all the resources available, such an assay can be developed in a couple of months. So I have not shown you those results yet, but we are progressing towards it. Basically, we have shown that we have now primers and other things available where we can show that in 30 minutes you can develop a coronavirus, you can detect a coronavirus, but now how to integrate it with the platform, that's taking a little bit time. And in terms of the sensitivity, this test which we are developing would be at least have about more than 90% sensitivity, which is much better than what we currently have in the market.
Great. Okay, I think these are pretty much most of the questions. I would like to wrap it up because we are a few minutes on top of the hour. So Dr. Asghar, I'd like to say thank you very much for your wonderful talk, very insightful, very beneficial for us to understand the work that is happening in your biomedical engineering and nanotechnology fields. So thank you very much. I'll end the call with this reminder to our audience that the STEM Matters is here to help, to provide service to the community. Our mission is to inspire students to excel in the areas of science, technology, engineering, and environment matters by nurturing their critical and analytical thinking skills. We coach and mentor middle and high schoolers and prepare them for the national and international STEM competitions. We'll be opening registration for the spring 2021 session next month. We look forward to having another batch of young students join us, and we'll help them sharpen their STEM skills and take them to the competitions. And hopefully they'll win. So you can find more information about us at www.untitled STEMmatters.org, STEMATTERS.org. And with that, Dr. Asghar, again, thank you. And thank you all for attending today's wonderful talk. I appreciate everybody's presence today. Have a great, wonderful rest of the day or evening. Goodbye.
So thank you, everyone. I think it was great talking to you guys. And I would love to actually talk more and have more time and answer those questions. So that were very interesting. And but I think based off the time constraint, we have to leave now. And but I think it was a very wonderful audience. So very, very energetic. Good to see you, everyone.
Great.
And thank you. Take care.
Take care, everybody.
Bye-bye for now.